So next in the topic of transport in plants, where we are dealing with the transport of water within the plant cells and tissues, we are going to deal with another term which is alike to transpiration, that is guttation. Now before we know what guttation is basically, I want you to know the difference between the three terms. First one that you have studied till now is the one that involved stomatas and was the necessary evil for plant responsible for conduction of sap, that was transpiration. The second one that we are going to take into consideration is bleeding, and the third one is guttation. Now, what you have to understand over here is that transpiration is the process in which the water is lost in the form of water vapors from stomatal pores. So, first point is that there would be water vapors, vaporized form of water would be there, and stomatas would be the openings which would be present in the leaf surfaces from where the water vapors would be getting out of the leaf surface into the atmosphere. Bleeding is the case where the plant tissue or the plant part undergoes an injury and the sap exudates out, okay, from the injured part. And last one is guttation, that is our topic of study in this lesson, which involves the giving out or the exudation of water in liquid form. That means you are going to find water droplets. Water is being given out in both the cases, transpiration and guttation, both are giving out water. The water vapors are in the case of transpiration, that means vaporized liquid would be present. You are not going to see water droplets. In case of guttation, water droplets would be present at the vein endings, especially vein endings of the leaves or other parts as well, and mind it would not be involving any kind of injury. And as I told you, in the bleeding case there is injury, because of which whatever cell sap or whatever sap is present inside the plant, that comes out. You must have seen that at times you break the leaves or you just tear apart the leaf from a plant and there is some milky substance that comes out, that is bleeding. But in the case of guttation, you would find this particular process, like transpiration goes on entire day, it is taking place, and because water vapors are involved which are gaseous and we cannot see, so we cannot differentiate whether transpiration is going on, but this guttation process is quite visible to our eyes. And early in the morning on a humid day, on a humid morning, early morning, what you're going to observe is that supposedly this is a leaf, there is a humid day, over here you're going to find tiny droplets of water, which would be water, and it is not completely water. There are certain amount of solutes inside this particular liquid, 6 to 7% of solutes are present, and this droplet of water that is formed at the vein ending, as I told you, it would be formed at the vein ending. The leaf is present, vein ending has a liquid droplet that exudes out early morning on a humid day. Okay, so early mornings, highly humid mornings, you are going to see the process of guttation taking place in many of the plants. Not all plants they show the process of guttation, but most of the plants they show the process of guttation in which the liquid is exudated out from the leaf endings, okay, vein endings. Now you have to keep in mind that these are not going to be in vapor form. These exudates at times, the water evaporates at times, and there is some powdery substance, the solutes they remain behind, and some whitish powdery substance that stays over there. And this guttation is a result of positive pressure. Now as you can see, this bleeding concept should be erased out of here, because this does not have much similarity, or the points of differences are much more It is a totally different concept, but these two concepts, that is transpiration and guttation, they run parallel, and then we are going to see that how it is different. So we find more points of differences and more parallel points. So that is why we are taking into consideration transpiration and guttation. Firstly, because transpiration is going to take place mostly in leaves, so as in the case of guttation, it takes place mostly in the leaves. Secondly, the transpiration involves the negative pull. So there is a negative pressure, that is transpiration pull, that is responsible for this process, while in the case of guttation, it is the positive pressure exerted by the root pressure. If you remember what root pressure was, it was the pressure that the water column was exerting. It was a positive pressure, and this process of guttation is the proof that tells that yes, there is root pressure inside the plants. So it proves that root pressure exists because of the positive pressure that is occurring, the exudates come out. Had it been negative, it wouldn't be present over here. So the transpiration is a negative pull, while guttation is a result of or resultant process that occurs due to positive pressure caused by the root pressure. Third thing is that we have water vapors over here, then we have water droplets. Like in the case of transpiration, we had stomatal pores. Over here, in the case of guttation, the pore that is responsible for exudation of the water droplet is known as hydatoid. So we have hydatoids, as is in the case of stomata. We have hydatoids in case of guttation, okay? While transpiration has stomata, guttation has hydatoids. Now these hydatoids are nothing but they are the arrangements of the cells at these points in a leaf. Now what it has is that it needs a little bit of description. You need to understand what hydatoid is actually all about. For getting rid of the water that is being pushed by a positive pressure, supposedly this is the leaf. Over here you would have a small pore that is known as water stoma, okay, or the water pore. Over here you find many non-chlorophyllous, that means colorless cells, known as epithelium cells. Please remember the names. These could be important from exam point of view. Water stoma is the pore surrounded by epithelium cells. There is the vascular bundle arrangement, and of course there would be parenchymatous mesophyll cells all around. So we are concerned about this part where we have water stoma, the pore, and the epithelium cells, otherwise known as transfer cells, which are present for the purpose of absorption of solutes. So when this positive pressure holding sap comes over here, there is a positive pressure, there is humidity, transpiration is almost nil. This positive pressure exerts, comes over here, the exudate reaches over here, it comes in contact with the epithelium cells. Epithelium cells absorb back whatever solutes are present, and from here the water pore, there is a tiny droplet of water that resides at the ending of the leaf. So this is the arrangement that shows what a hydatoid is all about. Like we had stomata, we had guard cells, we had subsidiary cells that made the entire stomatal arrangement. In the same way, the hydatoid has the water stoma or the pore, that is water pore, and the epithelium cells which surround that water pore for the absorption of the liquid solutes, not liquid solutes, the solutes that are present in the liquid, so that water droplet comes out at the vein ending. So this is hydatoid. I hope you can recall what we had studied in the stomatal aperture. They had two guard cells, kidney shaped guard cells in the case of dicot leaves and dumbbell shaped ones in monocot leaves. They had a stomatal pore, they had subsidiary cells. So it was a variation in epidermal arrangement. Over here you don't find the epidermal cells which are varied, it is the mesophyll cell itself which is varied in contact with the vascular bundles. We have the epithelium cells. They do not have much of chlorophyll. They are for absorbing back the solutes, and the rest arrangement has the water pore, which is responsible for giving out the water. So this is all about guttation that you have to understand. You have to understand what the process is all about. You have to know how it is different from bleeding, and the most important point is that because it involves both the process which is similar to the process of transpiration at the exudation level, and because also it involves the leaves, the main organ which is responsible for transpiration. So you must understand the characteristic differences between transpiration and guttation, and of course you need to understand what a hydrothode is all about with reference to the stomatal arrangement. So this is all you have to understand in the context of transport in plants. As we move further, you are going to see the process of photosynthesis, that how it occurs in the plants, and lastly you are going to see translocation. But for now we are done with the transport part, where we have discussed about transpiration, we have discussed about cohesion tension, we have discussed about few physical properties which are responsible for the various processes which are ultimately giving rise to transport of the sap within the plant, which were osmosis, plasmolysis, and rest many. So with this we come to the end of transport in plants.